Hello. So for the Cell Signaling Project, I chose to do breast cancer and estrogen. So this is my concept map. Since this video just has to be 3 minutes long, I'm not going to say this because this will take most of the time. So I'm just going to jump right into the presentation. Oops. Okay, so breast cancer facts. Cancer can occur in both men and women, but more common in women. 1 in 8 U.S. women will develop breast cancer. It is the most common cancer diagnosis in women in the United States. It most often begins with cells in milk-producing ducts. Also begin in the lobules, which are glandular tissue. 5 to 10% of breast cancers are linked to gene mutations. BRCA1 and BRCA2 are the most well-known genes in breast cancer, and mutations in the BRCA might lead to breast cancer. Estrogen. Estrogen are natural hormones developed in women for sexual development. Just like men, they have testosterone. Women, for sexual development, we use estrogen. Studies show that higher amount of estrogen in the blood is linked to an increase of breast cancer after or before menopause. Estrogen is produced by the ovaries in the first half of a woman's menstrual cycle. Estrogen enhances the development of milk ducts in the breast, and there are many types of breast cancers. You can have ER positive, ER negative. There's even something called like triple breast cancer. ER positive is when you have high levels of estrogen in your milk ducts in the breast. ER negative is when you actually don't have any, where estrogen is not the cause for breast cancer. So ER positive is when breast cancers that have estrogen receptors. They are also known as luminal A breast cancer. ER positive can be treated with hormone therapy drugs that lower estrogen levels or block estrogen receptors, and ER positive typically tends to grow more slower than hormone receptors. The ligand that binds the receptor is estrogen. The receptor only have one binding site, and the binding site is the endoplasmic reticulum, and the binding site is cytoplasmic. Second messages are involved, which is the cyclic AMP. The scaffold proteins that are involved is the MEK partner 1. Protein kinases that are involved are the SC kinase, mitogen activated protein kinase, which is also called MAPK, P13K, and protein kinase C pathways in the cytosol. So what protein kinases do, they phosphorylate the estrogen receptor alpha and inhibits dimerization in the absence of ligand. The G protein that is involved is called the GPR30, and protein tyrosine kinases are known to regulate downstream signaling pathways. So steps to breast cancer. First, the ligand activated ER alpha moves to the nucleus and binds to the CMP and stimulates gene transcription. Second step, estrogen binds to one of the ERs. It could either be estrogen alpha or estrogen beta. Estrogen alpha co-regulator, another word, A1B1, amplifies the BRAC1 gene. So once the BRAC1 gene is amplified and turned on, that is a possible indicator that breast cancer might be occurring. So ER alpha signaling stimulates the SC kinase, somatogen activated protein kinase, P13K, and protein kinase C pathways. ER kinase and protein kinase B phosphorylation controls breast cancer cell migration. So once there are mutations in those kinases, and once they are not available, but are in the body, that's when the breast cancer starts to migrate throughout the body, especially when the BRCA1 gene is amplified. The membrane-bound ERA is associated with growth factors of HER2, which promotes the growth of cancer cells. And HER2 is actually a gene that doctors know that you actually do have breast cancer when they spot the HER2. So estrogen alpha does also create HER2 within the body, and it leads to activation of MAPK. So for my normal signaling, as you can see, there isn't any ER alphas or ER betas in the nucleus, and I couldn't really find a normal signaling for breast cancer, so I just chose a diagram where estrogen is not the cause for breast cancer. So this is a ER negative diagram. And as you can see in the abnormal signaling pathway, ER alpha is at the bottom and it is leading to the breast cancer gene. And the breast cancer gene is also going towards ERA. So it's like a whole cycle where ER alpha starts the cycle and the BRAC ends it, and then it goes back to the ER alpha. Conclusion, the cell response is a nuclear response, and the gene that is generated is the BRC1 gene. So how is the signal terminated? It is terminated by blocking estrogen using hormone therapies, and it is terminated by the removal of the ligand so it can no longer access its receptor. And these are my references, and thank you so much.